Okay.
All right.
Yeah.
So I think it's 10:30. We can start. So hello, everybody, and welcome to the thesis defense of Gabriel Mangeat. I've known Gabriel since I started at Polytechnique. I think he's the last person in the lab that we actually interacted with as I joined. And it's really a pleasure to see him progress through his studies and reach this point of his career. Today he will be talking to us about the development of in vivo histology with quantitative magnetic resonance imaging to resolve fine neurodegenerative features. I would like to introduce the jury first. So here today is Julien's directeur de recherche, supervisor, Julien Cohen-Haddad, and also the co-director, Tobias Greinberg, who is joining us from Sweden. And I would also like to thank Delphine Perrier-Curnier for being a member on the jury and Christine Lucas-Tardif for being the external. And also I would like to introduce the representative du DES, Olivier Henry, and thank him for accepting this duty. We will start with Gabriel's presentation, and afterwards we'll do a round of questions from everybody on the jury. Then we'll ask the audience if there's questions, and we'll decide behind closed doors whether Gabriel passed. So with that being said, Gabriel, I would like to welcome you and ask you to present your thesis.
Thank you for the introduction, Nicolas. Can you hear me well?
Yes.
Okay, so thanks everyone for being here today. Thank you very much to the member of the jury for accepting to review my PhD work. So without waiting more, I will present you my PhD project. So here's the plan for this morning. We will start with a literature review about multiple sclerosis and quantitative MRI and histology. After this, we will pass through my research objectives and my different scientific contributions, which are 3 scientific articles published or submitted and some complementary work about histology. Then I will discuss this work and we will finish with some questions. So let's dive into it. Multiple sclerosis, or MS, is an autoimmune disease of the central nervous system affecting 2 to 3 million people in the world. It is mainly causing demyelination in the white and gray matter, inflammation, or even axonal loss. Myelin is a key component of the neuron and is responsible for the speed of neural communications. These pathological mechanisms are often translating to functional deficit in MS patients, such as paralysis, blindness, or chronic pain. So we can wonder what are the current scientific challenges of MS. The first one is that the causes of MS are still unknown. Is it genetic? Is it a virus? What triggers the disease is still unclear. Another challenge is the treatment of MS. As we can see on this chart, many new treatments arrived on the market in the recent years, which opens the door to a more personalized medicine. Today, thanks to this new treatment, the sooner is the diagnosis, the better is it for the patient outcome. MRI is the method of choice to study MS, whether it is for the diagnosis or to follow the progression of the disease. It provides non-invasive 3D images of the brain and is sensitive to several neurodegenerative mechanisms of MS. As we can see on the picture, clinical MRI can highlight dark or bright spots called lesions. For example, T1 weighted bright spots reflect area of active inflammation, while dark areas in T1 weighted images can be interpreted as permanent damages. However, clinical MRI has some limitation. What does it mean when an area is brighter than another, or how much signal is considered as bright? So hopefully there is another branch of MRI techniques called quantitative MRI, which is less used in clinic but allows to make quantitative measurements. So here is an example of quantitative MRI techniques. For instance, diffusion MRI can measure the orientation of the neural fibers. magnetization transfer or relaxometry techniques are sensitive to the composition of the microstructure. But despite being quantitative, those techniques might not be specific to one element of the microstructure. For example, the T2 star images will be sensitive to the concentration of myelin, but also to iron and maybe more. So different models can be used to gain in specificity to the composition of the microstructure. The tractography models aim to reconstruct the brain's neural network. The QMT approach is modeling the magnetization exchanges to recover the concentration of myelin, for example. The synthetic MRI models combine three relaxometry maps to estimate the myelin volume and the cellular volume in the brain. And during my master, I also proposed a model aiming to combine several metrics sensitive to myelin to extract one more specific metric. So once a model is made, we want to compare it with a reference, such as histological measurement. But the correlation between quantitative MRI and histology are often mitigated because of two things. First, there are biases in the models. They are just a simplification of the reality. And second, there are biases in the histology. For example, problems like inhomogeneous stain penetration or background absorption are hampering histological maps. And this is reducing their ability to be used as a reference gold standard. So to overcome the problem associated with staining, the techniques called stain-free histology have emerged in the past decade. The vibrational microscopy, such as CARS or SRS, can give specific maps of vibrational molecular bonds that can be tuned to image the myelin, for example. In addition, the hyperspectral autofluorescence microscopy uses the fact that different tissues will have different fluorescent spectral signatures that can be recovered by signal unmixing. So investigating the potential of these techniques to validate quantitative MRI could be of interest for the community. So this brings us to the objectives of my PhD. So my main research question was, what is the potential of multimodal quantitative MRI to decipher subtle neurodegenerative mechanisms of MS in a research and in a clinical setups? And what does it take to bring multimodal quantitative MRI to in vivo histology? So to answer it, I split my work in four different objectives. The first one was assess the sensitivity of research quantitative MRI metrics to detect subtle and early pathological changes in MS. The second one was assess the specificity of clinical quantitative MRI metrics to differentiate MS from a closed condition. The third objective was assess the repeatability and reproducibility of clinical quantitative MRI. And the fourth was assess the potential of stain-free histology to validate the accuracy of quantitative MRI. So pretty interesting program here. So the first study that I have made was related to the objective one. The title is Changes in Structural Network are Associated with Cortical Demyelination in Early Multiple Sclerosis. And the idea of the study was that since the structural network is composed of axons that connect with the cortex, we can wonder if there is an interplay between the cortical pathology and the axonal pathology. So in order to address this goal, I developed a method to combine ultra-high field quantitative metrics to study the cortical demyelination. And I used tractography techniques to detect changes in the structural network. So let's see our methods for the scans at 70. So 27 early MS patients and 18 healthy controls were scanned at 70. We acquired the T2 star maps at 0.5 millimeter isotropic. and the T1 maps using the MP2-RAGE sequence at 0.75 millimeter isotropic. Then, using FreeSurfer, I sampled the cortex at each volume at the mid-distance between the pial and the white matter surfaces. And this was done to minimize the partial volume effect. So the next step will be to combine those 70 maps to improve their specificity to myelin. So briefly, an independent component analysis is applied to the data in order to extract the myelin-specific signal shared by both metrics. The resulting composite map is called CME for combined myelin estimation. So now let's switch to our scans using the connectome scanner. So the same cohort of control and patient was scanned on a 3 Tesla connectome scanner. Then I used the software DSI Studio to obtain the tractography of each subject. And now we just need to properly register a cortical atlas to be able to extract the connectivity matrices. The brain connectivity toolbox was used to compute the graph matrix of interest. Here we computed the node strength, the local efficiency, and the clustering. So here are the results. These graphs are relating the gray matter and white matter changes. In abscisses, we can see the variation of cortical myelin estimation. And in ordinate, we have the variation of white matter graph theory matrix. So this result shows that both phenomena are not independent. It suggests an interplay between the variation of connectomics and the cortical pathology during the first stages of the disease. These graphs are showing the intensity of the pathology depending on the disease duration of the patient. So overall, we observe an increase of these metrics up to the 3rd year of the disease and a slight decrease after this. The trend of those results is consistent with a previous study who computed the same connectivity metrics on a larger cohort. But our study showed that this behavior is similar for both gray matter and white matter metrics. So in conclusion, our study highlights that the loss of cortical myelin and increase in connectivity appear to be synchronized, suggesting that the two phenomena might be linked. Future study might investigate whether there is a causal effect relation between the two processes. And finally, we show that the quantitative MRI techniques that we have used here are able to resolve fine neurodegenerative features occurring at the earliest stages of MS. So following the publication of the article, I have been invited to the French MS Society or RCEP Conference in Paris to discuss those interesting results. So for this first objective, I had access to state-of-the-art technology. As we saw, this permitted to resolve fine and subtle mechanisms. However, it comes with a long scan time, about 4 hours per subject. It is expensive to use, and because the technology is exclusive, it is quite hard for other centers to reproduce the work. So for my second objective, I focus on clinical quantitative MRI. More precisely, the protocol called synthetic MRI, which allows a lower resolution, which is maybe less informative, but which is very fast and accessible to most clinical centers. So discussing with my co-supervisor, Dr. Greenberg, I heard about a clinical challenge for which this clinical quantitative MRI could help. It is about a neurodegenerative disease called hdls, or hereditary diffuse leukoencephalopathy with spheroids. The disease is rare compared to MS and is unfortunately characterized by very similar radiological symptoms. so similar that that we can see current misdiagnosis between the two diseases. The problem is that the MS treatment on an HDLS patient will not help and only cause the side effects. So the question is, can a fast multimodal quantitative MRI technique like synthetic MRI be specific enough to distinguish those two conditions? So to answer this question, we hired and scanned 14 healthy controls, 14 MS patients, and 4 HDLS patients. Again, HDLS is a very rare condition. So all the subjects underwent a 7-minute using the multimodal sequence synthetic MRI. And then we extracted the quantitative T1, T2, PD, and myelin map from the synthetic MRI software. The whole classification pipeline that I used was quite simple. So the quantitative maps T1, T2, PD, and myelin were averaged in the different tissue classes, such as white matter or gray matter. And moreover, I computed the volume fraction of each tissues. So this step reduces all of the data of one subject to only 16 features. The features were then input in a model in order to output the diagnosis. So we tried several nonlinear models such as classification trees, but the most robust method here was shown to be a simple linear discriminant analysis. The next step will be the feature selection. So the goal is to find the best predictor to discriminate HDLS from MS. For this, I used Bootstrap random feature selection to determine the optimal number of features. So we can see on this graph here that with not enough feature 1 or 2, the prediction isn't good enough. With too many, the model overfits and leads to a lower cross-validation accuracy. And we reach an optimum with 5 to 11 features. The graph here is showing the importance of the features. Interestingly, the myelin map in the gray matter seems to have the highest contribution. And moreover, we notice that no feature is completely discarded. So all of them can contribute to the classification. Speaking of classification, here is an example of the result. The blue dots are HDLS and the orange-red are MS. And basically, each class is trained to stay on its side of the line. The graph at the middle is a combination or a bagging of several models. Overall, the confusion matrices show an impressive classification of the cross-validation data, 100%. But one point I still wanted to address is about the amount of data. So to better assess the stability of the model, I use a technique called artificial data augmentation. The idea is to create artificial data by adding different levels of noise on the original images. We can observe that with a noise of 3.3 and 5%, the results of the classification are not perfect, but still good enough to be used as an aid to the diagnosis. Why 3 or 5%? We will see later that it is close to the actual reproducibility of synthetic MRI. So in conclusion, we explored the feasibility to distinguish MS and HDLS from fast quantitative MRI. Based on this data, the answer is yes. Indeed, the method is fast and show very encouraging results. So the same approach could be used to try to distinguish other challenging differential diagnoses. One limitation is that the model relies on the absolute values of the quantification. So the repeatability of the quantitative measurement is critical here. And this limitation will be assessed in the next objective. The study here was published in the Journal of Neuroimaging. So the objective 3 is strongly related to the objective 2. The goal was to assess if the algorithm presented in the objective 2 can be generalized to different MRI platforms. So during a 3-month internship at the Karolinska Institute in Stockholm, Sweden, we have made hundreds of scans to answer this question. And yes, we had hamlets in the MRI room because some wings of the hospital was still under construction. So more specifically, I aim to assess how repeatable and reproducible are the quantitative measurements of the clinical sequence synthetic MRI. Is it possible to compare quantitative MRI across centers? So we try to answer this question by adding the most variability to the data by using 9 scanners from 3 vendors at 2 field strengths. In terms of methodology, each subject was scanned 5 times per scanner, 3 times without reposition and 2 times with reposition, for a total of 165 scans. We use the default sequence parameters for every scanner. Then the software synthetic MRI was used to calculate the quantitative T1, T2, PD maps and the myelin estimation remedy, as well as the 2 masks, white matter and gray matter. Here is an example of the data. So these are the T1 maps of one subject for every scanner and every rescans. Visually, we observe a good consistency between the maps. Here are the graphs comparing all of those quantitative maps in the white matter. So for each healthy controls, we expect to have the same measurement. The purple and red line are showing the 1% and 5% variation to the mean measurement. And overall, we observe that all measurements seems to be within a 5% precision, except for the T2 acquisitions. Here are the actual repeatability, so error per scanner, and reproducibility, error inter-scanner. For example, for the T1 in the white matter, I have 697 milliseconds, more or less 8.7. So this number means that if I take the subject out of the scanner and put him back in, we have 95% chance that the value of the measurement will be comprised within the repeatability error. For the reproducibility, it's even more interesting. It means that if I take the subject out of the scanner and put him back in another scanner, I have 95% chance that the result will be comprised within the reproducibility error. Here is what it means in terms of coefficient of variation in percentage. So we observe a good intra-scanner repeatability and a decent reproducibility. Almost always less than 5%, except for the myelin in the gray matter. So in this study, we show that using a single scanner, there is no problem in relying on the absolute values of synthetic MRI. There is more variability across scanners, in average 5.5%, which is still acceptable for many applications. So this opens the door to multicentric clinical studies. In addition, a calibration per scanner, for example, with a phantom, could be used to enhance the reproducibility. So here we just saw that it is possible to assess the repeatability of quantitative MRI, or how much does it agree with itself. But there is still something missing. Can we use a different method to make the same measurement? For example, in the last chapter, we used the myelin model from synthetic MRI to measure the myelin fraction of the brain. We have seen that it is repeatable, but is it accurate? Will I have the same value if I make the same measurement with a model based on QMT, for example? So to answer this question, we need a measurement of reference that we assume to be true. Then we can use it to compare different methods. For T1, T2, and PD, we have a device called a phantom, which contains small spheres of liquid with known T1, T2, and PD. We just need to scan it and compare. Here is an idea of what I had scanning a phantom with 2 different scanners with the synthetic MRI sequence. T1 is pretty accurate, always less than 5%. T2 is less accurate, depending on the range. And PD, despite it stays precise, has an obvious problem of accuracy. But now, what about microstructural quantities like myelin? Well, we don't have such phantom, and the method to obtain the gold standard is more challenging. we need to use histology to get the reference gold standard. So as we saw earlier, stain-free technique could be of interest. The goal of this fourth objective was to explore the potential of stain-free techniques to validate quantitative MRI and design and test a validation protocol. So the study here was presented at the conference ISMRM 2019. And here I investigated the technique called hyperspectral autofluorescence. So basically, a UV laser is sent to a sample using a confocal scanning microscope. For every pixel of the image, we record the emission spectrum of the autofluorescent signal. Then we can unmix the signal to separate the different tissue maps. Here on the right is an idea of the optical setup that I use based on a commercial Olympus platform. Here is the protocol that we developed and used for this study. So first, we have made in vivo scans of a pig to image its spinal cord with different quantitative MRI techniques. The pig was then euthanized and its spinal cord was extracted, inserted in a fixative solution, and put back in the MRI in a custom-made coil designed for this purpose. And the same quantitative MRI measures were repeated with this setup. Then the samples were sliced and mounted on slab for histology. Here are the images that we obtained for the in vivo and ex vivo quantitative scans. We can observe a shift between in vivo and ex vivo, which is likely due to the fixation and changes in temperature. Here is an idea of the histology results. On the raw images, we observe some tiling artifacts that were corrected. And then to unmix the individual tissues, I tried two different approaches. First, the unsupervised approach, using a PCA to separate the orthogonal sources of signal in the spectra, and a supervised approach. So some slices were stained with a myelin stain called black gold, and a machine learning approach was used to assess the best combination of hyperspectral channels to predict the myelin. We can see that the unsupervised tissue estimation correlates well with most of the quantitative MRI. For example, PC2 correlates with MTR, MTSAT, and T1 with an error around 0.6, which is good, but to me, not yet enough to validate the accuracy of quantitative MRI. The supervised methods show fairly low correlation with both the MRI and hyperspectral histology. So after some investigation, we saw that the black gold staining did not perform well, and some stain agglomeration was present, and an inhomogeneous stain penetration occurred. So with that in hand, I choose to investigate another staining method called the Nile red, to be able to build a better myelin model from the stain-free images. So using the Nile red fluorescent myelin stain, the images were very clear, and the contrast between myelin and background was really sharp. Thanks to the high sensitivity and the high quantum efficiency of the stain, I could acquire images more than 20 times faster than with the hyperspectral protocol. Concerning the specificity, we can see that other structures than myelin seems to be highlighted, but they were mostly seen in the gray matter. And keep in mind that the goal was just to train a model to find the optimal combination of hyperspectral channels. So despite very promising images, I unfortunately had to stop those experiments last year due to the lockdown. Nevertheless, we can draw interesting conclusions for this, from this fourth objective. So first, validating the accuracy of quantitative MRI with a stain-free protocol is possible, but rather challenging. Secondly, using the spinal cord tissue is the histology part, but makes the MRI imaging more noisy and challenging. The hyperspectral autofluorescence technique is natively sensitive to the myelin when a 405 nanometers laser is used, but some signal processing is needed to gain in specificity. To gain in imaging speed, we could use low resolution full field of view images to compare with quantitative MRI, and small high resolution images to quantify the actual sensitivity and specificity of the technique. So it is now time to conclude. So in this presentation, we have seen that first, combining state-of-the-art quantitative MRI techniques can highlight subtle and early neurodegenerative mechanisms of MS. Also, we've seen that fast clinical quantitative MRI can help for difficult differential diagnosis. In the third objective, I highlighted that knowing the repeatability and reproducibility of synthetic MRI allows to set the resolution limit for multicentric protocol that relies on the quantitative images. Finally, using label-free histology to calibrate the accuracy of quantitative MRI is possible, but more development are needed to quantify the gain compared to the staining approaches. So now I'd like to take 1 minute to talk about my journey in research. So I started academic research in MRI during a back internship 7 years ago. From then, I have contributed to 11 journal articles, 25 conference publications, and of course, a lot of unpublished projects. With 300 citations, it seems that this work has been in some way useful for the community. But sometimes I kept wondering, okay, who will be using this? How can it be more useful? In parallel, 3 years ago, I had the chance to start a project with a lab mate of Neuropoly about a sustainable technology. The project has grown and we are today a small company of 15 employees. The point is that the synergy between both projects was very good. I had to combine the need of always doing innovative things as we have to do in the academy, but doing them quickly for a well-defined short-term purpose as we need to do in the industry. So this helped me to reorient my research objectives sometimes to be more and more practical and closer to the daily problems of patients and doctors. So I would like to thank the member of the jury for the time taken to review my work. Thank you to all of my lab mates for your support and those wonderful moments in research. Thank you, Tobias and Julien, for all of the experience you have shared with me and all of those opportunities to travel the world to present our research, like here in Hawaii for ISMRM. And a word for those who joined the lab during the pandemic. Hang on, life in research is cool. I'm sure it will be back to normal soon. That's the time for the questions.
Excellent.